a question for Chris, actually. Are you able to talk in a little more about the process of how a drug is defined as resistant via whole genome sequencing, the reliability, and which result to believe if, for example, pyrazinamide phenotypic drug susceptibility testing suggests susceptibility and whole genome sequencing raises the possibility of resistance?
Okay, I think I'm on the air.
Yeah, we can hear.
Yeah, so for some mutations, there's a good track record of association with between a particular mutation and resistance, and the RPOB rifampin resistance determining region is a good example of that. It's backed up by in vitro drug susceptibility testing, but as I alluded in my talk, there can also be some mutations where the phenotypic susceptibility hasn't been reported as resistant, but you can get treatment failure. So for some of those mutations, we know that treatment failure can result, and it's a little bit method dependent on whether your susceptibility data tells you R or S. In the WHO catalogue, they started, they had 38,000 isolates to look at. So they basically used putative mutations where there was some track record of feasibility, probability that they were associated with resistance. It means that there may have been some rare mutations which had been discarded, but if they had to go through a process where they discarded highly improbable associated mutations, these were called null mutations. Then they looked at anything that was synonymous. So in other words, a mutation that while changes the base pair, it doesn't actually change the amino acid sequence, and therefore not unlikely to confer resistance. And so they tried to then get a process where they got down to what they called solos. In other words, a single mutation associated with resistance. And then they looked at how often did that mutation, if you've got, say you've got 5,000 isoniazid resistant isolates, how often was a particular mutation found in the resistant ones, and how often was it found in the susceptible ones by the laboratory testing? And bear in mind, this was a data set made up of contributions from different sources and some variability in who was doing the drug susceptibility testing and how it's done. So basically they tried to arrive at a probability or an odds ratio that a particular mutation was associated with susceptibility. There will likely, I think they're already working on version 2 of that document, so there will likely be new things found, new adjustments. For instance, with bedaquiline and clofazimine, RV0678, which is a gene associated with resistance, it didn't come up in the data set. That was a bit of a surprise, but it just was underrepresented in the data set. And the MIC creep that you see with the mutations in that gene are often below what we've decided the critical concentration is. So therefore, what you decide the critical concentration is as your sort of gold standard is fairly important in deciding which is chicken and which is egg. As far as pyrazinamide goes, pyrazinamide is tricky. It's probably, I think we would probably find false resistance more than false susceptibility, and it's a little bit prone to inoculum problems. So one of the approaches, if you're getting discordant results, is to reduce the concentration of the inoculum, and often that will solve the problem. Clearly, you can use that catalogue to look up particular mutations, and sometimes, yep, you've got a discrepancy. The catalogue says it's not associated with resistance, I think the example you gave was phenotypically susceptible but genotypically resistant, and then sometimes you can't resolve it. But those sort of data tables from the WHO catalogue, and you can find similar things in, say, the ReSeq database, will tell you how many times that mutation is associated with susceptibility and how many times associated with resistance. So that's the long-winded answer.
Okay, thanks, Chris. Andrew has his hand up.
Oh yeah, thanks. Yeah, Willem, that's great. With your linezolid saliva assay, if you like, so are you imagining that will be done as a trough level, and does it correlate with AUC, or will it be multiple samples?
Yeah, so our saliva assay with the mobile device, I think the trough level is a little bit of a risk. And the situation is that with saliva point of care testing, you're not able to clean up your sample. So there is some interference at the lower level of linezolid with other TB drugs. That can be overcome when we are looking at the peak level of linezolid, because then the interference is negligible. So I think for HPLC UV or LCMS, I think it's fine. You can have a trough level for routine care. I think for studies, it's not sufficient because there is still some error in having only a trough sample. So I think we should try to distinguish what is suitable for a clinical trial and what is suitable for clinical practice.
And that leads into my second question. I mean, as you pointed out, for most of the TB drugs, AUC is the key parameter we look at. So in clinical practice, as opposed to clinical trials, sometimes I've done the AUC for rifampin and isoniazid, and sometimes it's been a peak. And the question is, well, what would you do in your practice if you're worried that somebody might be at risk for low levels or toxic levels? What would be your limited sampling strategy that you'd recommend in clinical practice?
So for clinical practice, I would not aim for a trough sample if we're looking at drug susceptible TB, because in most situations will all be very, very low. So I would at least take 2 samples post dosage, and whether that is 2 hours and 4 hours, 2 hours and 6 hours, something like that, it will give you a fairly good idea, ballpark figure what AUC could be, especially if you can combine it with Bayesian software. Yeah, and then if you're in the luxurious position, as I was in my previous TB clinic, we did always a full PK curves when we had special cases, and that of course resulted in a lot of case reports and clinical studies, and it's very informative. Yeah.
And I guess, I mean, one of the things for me when I was looking at, what's the point of doing TDM in a country like Australia where you have high cure rates? There does seem to be, in terms of time to clinical to negative cultures, that getting the dosing right early on does seem to decrease the amount of time that people would probably be in hospital. These patients who had TDM done, the ones I discussed, it was like week 8 or 10, and probably the boat has already sailed. I think for those people, they probably could have had TDM done earlier. It's a learning point for all of us. And that may have then influenced their, the course of their disease a bit more than doing it, months into treatment for fully susceptible.
Yeah, and I think you're perfectly spot on with this. I think Chuck Peloquin has a very nice slide that he frequently uses, and it is about the US program for TB. And that shows that over 90% of the patients complete their 6 months treatment in 12 months. So although overall cure rate looks nice, if you look to what's happening to get there, it's a lot of work, toxicity, all those other issues, and likely TDM can help to prevent all those issues.
I think Chris has a
Chris
question.
has his hand up.
Yeah, thanks. Sorry, I was couldn't work out how to put the question in the chat for some reason. Look, my question is also to Jan Willem. One of the assays that Stephen Gillespie's group in the UK have developed is this Mycobacterial load assay, and it uses sputum to do a reverse transcriptase quantitative PCR targeting the ribosomal RNA gene, or back to the ribosomal, it's not the gene, it's the actual ribosomal RNA, and which of course there's multiple copies, and it does basically, as you treat people, the MBLA comes down, and you can measure it much more accurately than, say, smear positivity or culture conversion. Are you aware of anyone using that assay for a short-term endpoint in PKPD TDM trials, or do you think that would be a useful thing to explore?
I think it's a very useful tool to explore because, as you mentioned, it's more precise and less laborious than all the traditional cultures and plating and replating, etc. I think that in the trials done by Stephen, also including drug exposure, likely they will do the double and look at exposure and at this assay. But I'm not aware of anyone doing particular TDM studies. So as I mentioned in my talk, exposure is often included, but only as a, let's say, observation, but not really used as a opportunity to make dose adjustments during the trial.
Oh, if we set it up, I'll let you know.
Yes.
I think they're going to commercialize it, which is why there's a bit of a not too much in the last year being published, but it looks very promising to me as a monitoring tool.
Yeah, and it is very exciting. Exactly.
Thank you.
There is another question for Andrew or any of the speakers. How difficult is it for a TB lab to start doing TB/NTM?
Yeah, thanks. So that was James, and James kindly shared one of his slides in my talk. So, I mean, to best of my knowledge, Queensland Pathology is the only lab in Australia which provides, NADA accredited TDM assays for TB. So our lab already have a great expertise in TDM, so they've got that skill set. When I asked them, it probably costs about $4,000 per assay to make up each drug. And what they did was, when I said $100 per test, they have two panels, if you like, for hydrophobic and hydrophilic drugs. So $100 will basically give you all 4 drugs. And so they were able to do that. It took them time, but that wasn't hard for them to do. The one they struggled with was imipenem, which we use for non-tuberculous mycobacteria, which initially they vouched for and was accredited, but then they feel it's unstable. But I think if your lab's already doing TDM and developing their own drug assays, it's possible for them to do. The question is in Australia and New Zealand, we only do 1 run a week because there's just not enough demand to justify probably lots of labs doing it. And that is something which also influences our turnaround times and things. So yeah, it's certainly possible if you've got the time and expertise, and it's not a huge cost in the overall picture.
Yeah.
But there doesn't seem to be many labs around the world who publish their work. I'm not sure, Willem, I know the Netherlands, your previous home, has done probably the lion's share of this, but globally, do you think there's many labs who provide TDM, many countries?
I think there are more labs doing TDM than we are aware of, because not everybody publishes their assays. And also the problem is who is interested in publishing the 20th rifampicin assay. I think in many countries, China, India, all the high burden countries, they have assays, but of course they struggle with funding for that particular work, and also with turnaround times. So I think there is a true need for point of care assays where we can cheaply and easily screen patients at the bedside, and only when that screening assay says there is an issue, then we could consider a more full-blown TDM measures, either using a dry blood spot or something else to get it to that reference laboratory.
And Pathology Queensland, we do quality control with a Dutch laboratory for our TB assays as well. So we have that external link, which we're really grateful for. There was a question which I think has been taken off. Yeah,
a question just disappeared.
Yeah. I think we're related to the highest dose of rifampicin that's ever been used. So I think if we think about beyond dose and think about exposure, so there's now doses people have been trying, 3 times 30 milligrams a kilo of rifampicin. So probably 3 times our starting dose, particularly say, TB meningitis and things like that. So certainly, and those safety studies are quite good. So you can probably use much higher doses quite safely.
Yeah, sorry, that was me provisionally answering the question, saying it's for others to answer, and then as soon as I did, it disappeared from the Q&A, but yeah, I think Martin Bormann has done trial work with, 35 mg per kilogram.
Yeah.
I think once you get to that level, you start getting a bit more side effects, but that, and some of the trial work is only like first 8 weeks, but in general, the principle is that A, we probably don't have the critical concentration right for rifampicin, even with dropping it to 0.5, and B, the original dose of rifampicin was decided largely on cost, as I understand it, because it was an expensive new drug of a new class. It was a rifamycin, and it was sought 600 milligrams a day. Seems about right. I think that's as scientific as it got, but I could be wrong.
Oh, that's perfectly spot on. It was a little bit of argy-bargy between cost and effectiveness, and it was not toxic at that concentration. So that was how they marketed it. Currently, indeed, dosages up to 40 milligrams are fairly well tolerated. But of course, 30 milligrams per kilogram is a lot of capsules or tablets or whatever. So I think if we could develop a more powerful rifamycin that takes less tablets, would definitely benefit the treatment. Yeah.
Rifampin might have taken away from that argument.
Yeah, but that is not the winner of rifampicin, as I'm told.
All right. Well, look, I think that's I think I'm just taking over now from Jason Roberts as he just to close and just thank everybody for their talks, to Alice and Holly for sharing so well. And I always learn a lot from doing these talks, and grateful for Chris and Willem's mentorship over the years for a lot of these difficult problems. I'm not sure we thank our sponsors. Thanks very much. You can see them down the bottom. I'm really grateful for their support for this educational series. And you can see CR Reduce, which is for UQCCR. We've got a number of masterclasses throughout the year. So they're also recorded. They're available also on YouTube. So thanks, everyone. Hopefully you guys can all talk about this. And the TB group in Australia, it's not that big. So we're just looking for people to collaborate with and just to learn from and share your clinical cases. So thank you, everyone.
Thank you all.
Thank you, everyone.
Have a good evening.